everyone, my name is Avery and welcome to the iGEM Breaking Down Synthetic Biology crash course. Today we will be talking about promoters. In a previous video, we discussed the different parts of a plasmid. In this video, we are going to focus on one specific part that can be included in a plasmid, the promoter. Promoters are sequences of DNA that initiate and control the transcription of genes. In order to start transcription, RNA polymerase and transcription factors bind to promoters. There are two different types of promoters in both prokaryotes and eukaryotes: constitutive and inducible promoters. Constitutive promoters are promoters that are always turned on, meaning that RNA polymerase is normally always binding to these promoters and transcribing their genes. Constitutive promoters can be controlled by the ability of RNA polymerase to bind to the promoter. If the promoter has a weak binding affinity for RNA polymerase, it will limit the transcription of the gene. While constitutive promoters are always active, inducible promoters are only active in specific conditions. Some of these conditions include the presence of chemical agents, light, and even temperature. While the presence of certain conditions can switch the promoters on or off, often these promoters work on more of a sliding scale. For example, if there's a promoter that responds to the presence of lactose, transcription will increase as the presence of lactose increases. Inducible promoters are extremely important for the functions of cells. They allow a cell to create proteins only if they are needed, saving valuable energy and resources. For example, let's consider the lac operon. Normally, a repressor is attached to the lac operon. However, in the presence of allolactose, the repressor detaches and allows transcription of lactose-digesting proteins. Within the lac operon, E. coli do not need to produce proteins for lactose digestion if there's no lactose in the surrounding environment. If they created these proteins, they would be wasting valuable energy and resources that could have been devoted to accomplishing something more productive. Inducible promoters can be activated by 4 different kinds of control: positive inducible, positive repressible, negative inducible, and negative repressible. In positive inducible control, an activator protein is required to bind to DNA to initiate transcription. However, this activator protein is only able to bind to DNA when an inducer molecule is present. An example of this control is the TET-on system. In this system, an activator binds to DNA in the presence of tetracycline, starting transcription. In positive repressible control, the activator protein is usually bound to its DNA segment. When certain molecules called inhibitors are present, the activator protein is no longer able to bind to the DNA, halting transcription. An example of this kind of control can be seen in the TET-off promoter. Normally, a trans activator binds to the promoter, causing transcription. However, in the presence of tetracycline, the activator is removed and transcription stops. Negative inducible control takes place when a ligand, called the inducer, binds to the operator, causing it to be released from the operator, which activates transcription. This type of control is exhibited by the lac operon mentioned earlier in this video. Normally, there's a repressor bound to the lac operator. However, in the presence of allolactose, the repressor detaches from the DNA and allows the gene to be transcribed. Negative repressible control occurs when a molecule binds to the repressor, causing a shape change in the repressor. This shape change allows the repressor to bind to the operator, stopping transcription. This type of control is exhibited in the trip operon. When tryptophan is present, it binds to the repressor, allowing it to bind to the DNA, halting transcription. In addition to categorizing promoters as inducible and constitutive, promoters can also be classified as endogenous, heterologous, and synthetic. Promoters that are found naturally in an organism are called endogenous promoters. For example, the lac operon is endogenous to E. coli. When a promoter from one organism is inserted into another organism which naturally lacks this promoter, the promoter is said to be expressed heterologously, as is with any genetic sequence. Heterologous promoters are very useful in fields such as genetic engineering and synthetic biology. Synthetic promoters are new sequences of DNA that do not exist naturally, but are designed using different promoter fragments. This may create stronger promoters than ones that are found naturally and are very useful in synthetic biology. That being said, promoters do exhibit some specificity to the host organism. Bacterial promoters typically work in prokaryotic cells and generally in the same species or ones that are closely related to the species from which the promoter was obtained. This specificity stems from the varying transcription methods and parts between organisms. The RNA polymerases that attach to promoters for transcription initiation also exhibit a very significant degree of specificity towards the promoter region. When a plasmid is designed, promoters are chosen keeping their specific nature in mind. Due to their ability to act heterologously, scientists have been able to use promoters to their advantage in the lab. Inducible promoters play an important role in synthetic biology because they allow scientists to turn genes on and off. Often scientists will create synthetic molecules that mimic natural inducers. Using these molecules, scientists can initiate transcription without the natural inducer. Due to their sensitivity to inducers, inducible promoters can act as indicators of certain conditions in their surrounding environment. Scientists at Harvard University use this quality of inducible promoters to help detect and remove mercury from water sources, using the MER-R promoter. Usually, a repressor binds to the MER-R operator, halting transcription. However, in the presence of mercury, the repressor undergoes a conformational change and is released from the operator, illustrating negative inducible control. These scientists took the system and edited it to express the Curli operon when exposed to mercury. The Curli operon leads to the production of Curli, or protein fibers. These protein fibers are able to trap mercury molecules and remove them from an aqueous environment. Constitutive promoters have also played a valuable role in synthetic biology. They allow scientists to constantly produce materials regardless of the environmental conditions. For example, researchers have engineered bacteria to produce beta-carotene, which can be used to make vitamin A. These scientists transferred 3 genes involved in the synthesis of beta-carotene into this bacteria. In the bacteria, the transcription of all 3 of these genes is controlled by a single promoter, the TEF1 promoter. The TEF1 promoter is one of the most constitutive promoters, allowing almost constant production of beta-carotene. Using this promoter, scientists were able to greatly increase the amount of beta-carotene produced, decreasing production costs and increasing production efficiency. Promoters play a crucial role in the transcription of DNA. They provide RNA polymerase with a place to bind to DNA to initiate transcription. While they may seem complex, this complexity allows them to operate in all the different ways that your cells need. Thank you all for watching our video. We hope it promoted you to have an interest in promoters.